Hi, my name is Hajira Farah, and I am a senior at the University of Washington, majoring in microbiology, and this is my talk, Soft Agar Motility Analysis Reveals Heterogeneity in Motility and Chemotaxis During Chronic Stomach Colonization by Helicobacter pylori.
So just a little background on H. pylori. Helicobacter pylori is a gram-negative, helical rod-shaped bacterial pathogen that colonizes the stomach in more than half of the world's population. Most people do not show symptoms. However, complications can arise in some, and this includes ulcers, gastric cancer, and gastric malt lymphoma. Although more than half of the world's population are infected with this bacteria, prevalence can vary from region to region, with most developed countries reporting a low prevalence of H. pylori infection. And H. pylori infection is the strongest known risk factor for gastric cancer, which is the third leading cause of cancer-related deaths worldwide. So it's crucial to study the persistence and pathogenesis mechanisms of this bacteria in order to reduce the risk of H. pylori-induced gastric cancer. So the acidic environment of the stomach poses a challenge for bacterial colonization, and H. pylori is not an acidophile. And so this raises the question, how is it able to infect and persist in the stomach? And this is due to a few factors. Urease-dependent ammonia production neutralizes the pH, and chemotaxis-driven motility allows the bacteria to navigate down the acidic gradient towards the neutral gastric mucosa. Its helical shape is thought to enhance initial colonization through a corkscrew mechanism. And once H. pylori colonizes the gastric mucosa, it employs strategies to evade the host immune system, allowing it to persist in the stomach. Previous findings in our lab have shown that these phenotypes can vary within a single host over time.
And these findings draw from a study of a collection of isolates taken from a male patient who visited a clinic in 1994 and was diagnosed with a duodenal ulcer. An antral biopsy was taken and 12 isolates were collected. He refused treatment for the H. pylori infection at the time, but returned 6 years later, and another biopsy was taken, this time 24 isolates from 3 distinct regions, the duodenum, antrum, and corpus. And as you can see in this figure, the infection spread from the initial site to other regions of the stomach. So to understand how these bacteria genetically diversify over time, we're using the NextStrain program, which you may be familiar with. Dr. Bedford at the Fred Hutch, who helped design this program, has been using it to map the phylogeny of SARS-CoV-2. But here we're using it to look at how these bacteria are related. This tree displays the 39 isolates from the male patient grouped by genetic similarity. There are 4 subgroups, 2 from the first time point and 2 from the later time point. And as you can see, this tree shows genetic divergence over time. This divergence is reflected in observed phenotypes such as cell shape, motility, and chemotaxis, and may be reflective of adaptations of the bacterial population as it expands to colonize different regions of the stomach. So, as I was mentioning before, previous findings in our lab have shown that cell shape and other phenotypes vary over time. This is the same tree, but annotated with cell shape clusters. As you can see, isolates from the first time point all have a long pitch helical cell shape. Isolates from subgroup 2A all have a short pitch helical cell shape, and most of the isolates from subgroup 2B have a straight rod cell shape. And so Laura, the graduate student that I now work with, who was studying this cell shape phenotype, noticed as she looked at some of these strains under the scope that some were not as motile as others, and so we decided to test the swimming motility of these isolates using soft agar assays. A soft agar assay is a media that is commonly used to test the swimming motility and chemotaxis of bacteria. The method involves inoculating bacteria into a nutrient-rich media. Cells that are motile are able to migrate outwards from the point of inoculation, and this results in a visible motility halo, which is what we see here on this plate. This image, on the other hand, shows a non-motile mutant, and as you can see, at the point of inoculation, there is growth, but there is no motility halo. And so in our motility experiments, we inoculate soft agar plates with the H. pylori strains using sterilized straight pins, and then imaged the plates 5 days after inoculating. And we used Fiji, an image processing program, to measure the halo diameters and normalized our data sets to our positive control, J99, and MOTB here served as our negative control. This is showing the tree annotated with the average motility halos measured for all of the isolates. The warm hues represent motility halo diameters ranging from 0.7 to 1, and the colder hues represent smaller motility halos. And what you notice is that isolates from subgroup 1A collectively display higher motility compared to isolates from the later time point. Given the role that cell shape plays in regulating motility in viscous media, we thought it would be interesting to see if there are any trends observed. This is showing the same information plotted on that last tree, but here the isolates are grouped by cell shape. And so we find that isolates with a long pitch helical cell shape collectively show higher motility compared to the straight rod and short pitch helical isolates. However, as you can see, there is heterogeneity in the helical isolates. So I noticed as I was measuring halos that the isolates form rings in soft agar, which is characteristic of chemotactic bacteria. And bacterial chemotaxis is basically the directed movement of cells towards or away from the chemical gradient established as cells consume nutrients. And this depends on whether the compound it's sensing is an attractant or repellent. For flagellated cells, this translates to the regulation of flagellar rotation, either in the clockwise or counterclockwise direction, where CW is random tumbling and CCW is straight swimming. And so this method allows a bacteria to migrate towards optimum conditions. So the rings that you see here are formed as the bacteria consume preferred nutrients. What we noticed was that these isolates actually display variable chemotactic patterns. Some strains form 3 rings, some form 2 rings. As you move out from the point of inoculum, you'll see that there is a dark, more transparent halo followed by a more dense-looking ring on the outer edge. And some do not form rings at all, indicating that these strains are non-chemotactic. So looking again at the tree annotated with the chemotaxis phenotype, the blue color represents strains that do not form chemotactic rings. Yellow is the 2-ring forming isolates, and orange is the 3-ring forming isolates. And so you'll notice that chemotaxis is lost in isolates from the most recent time point, notably in subgroup 2A. So considering the variable patterns and halo sizes that these isolates form in soft agar, we wondered if perhaps differences in flagella number across these isolates might account for the heterogeneity that we observe in these phenotypes. So we did a flagella count for select strains that are either outliers or do not form chemotactic rings. So if you look at these electron micrograph images here on the right, the topmost picture here shows a cell with no flagella, this one shows a cell with 3, and this one shows a cell with 6. And so for the flagella count, I looked at a sample of cells for each of these isolates and counted the number of flagella on each of the cells. And looking at this plot, you see for C11, the vast majority of cells do not have flagella. Other strains display different patterns, with some strains like J99 and C2 having a median flagella number of 3, while others like A8, D1, A3, and A1 show a similar distribution, with the majority of cells having 4 or more flagella. And so, although these findings are interesting and informative, the interplay between cell shape, chemotaxis, and motility is complex, making it hard to draw conclusions from these findings. So in summary, our motility experiments show that there is heterogeneity in motility and chemotaxis phenotypes across these isolates, and this reflects the genetic divergence that occurred over time within a single host. For future directions, I plan on comparing SNPs in flagellar and chemotaxis genes to determine if there is a genetic basis for the differences that we observe in these phenotypes, to complete a chemotactic ring ratio measurement for the chemotactic ring forming strains, and to do a flagellar count for other strains. And so studying how these cell shape, motility, and chemotaxis phenotypes evolve in a host over time will hopefully provide insight into the persistence and pathogenicity mechanisms of H. pylori. Special thanks to the Salama Lab and to Laura, who has been a wonderful mentor to me this past year and without whom this project would not be possible.